Okay, so we're going to continue with our metabolism. So we were talking about the protein breakdown and how proteins can be utilized in terms of ATP production. We started talking about proteins, which is basically made up of amino acids. Amino acids are carbon. I'm going to redraw this again for you guys. The hydrogen, the R group. One side is carboxyl group or COOH, and the other side is NH2 groups, which is the amine group. So when you are undergoing the utilization of amino acids for production of ATP, you need to get rid of this amine group here, and that happens through a process called oxidative deamination. So again, here in this case, this is really important. What you actually produce is you take the amino acid, and by removing the amine group, you're producing ammonia as a byproduct. You also will have NADH produced, which can be used in terms of electron transport chain. And you're also producing the acetyl-CoA, which again can go through the Krebs cycle and eventually electron transport chain. So let's look at each of these on their own. So ammonia can actually be combined with CO2, and that becomes basically urea. So if you combine CO2 and ammonia, it becomes urea, and urea is basically a common waste product that can be removed through urine. NADH can eventually go to your electron transport chain and help you produce your ATP. And then acetyl-CoA, kind of same thing as we described for the fats, right? So it can go and I'm going to do a side arrow for it. And a kind of a double arrow we did before. So you can go through the process of ketogenesis and produce basically what we identify as ketones, right? When there is requirement for transportation. And the acetyl-CoA can eventually be used again in your Krebs cycle. And then again, transfer through electron transport chain and ATP for production for those high energy electron carriers. So right here, this is what you need to know for proteins. Now do keep in mind, does it apply for every protein, every amino acid that are found within the protein? The answer is no. Some of them are undergoing different changes. But for what you guys need to know for your understanding of the class, I need you to understand how glucose is utilized, going through the process of glycolysis, acetyl-CoA production, Krebs cycle, and electron transport chain. Then how does these processes we're describing, it's utilized when you're catalyzing your fats or your triglyceride? What part of the process does it enter? What are the proper terminology for them? Apply the same concept in terms of proteins and utilization of the amino acids to produce ATP. And again, remember that the oxidative deamination produce that ammonia byproduct, which is very harmful, and therefore your body converts that to urea by combining it with CO2. Now, one of the last concepts that we're going to talk about is the 3 phases of starvation. So starvation has 3 phases, first, second, and third phase. First phase usually happens within a few hours after you haven't ate anything. This is where you tap into the storage of complex carbohydrates like glycogen and utilize that during the fasting process, basically. This is really common, so a lot of people actually undergo this. There's a lot of benefits into undergoing this kind of a starvation phase where you don't eat for quite a few hours, especially if you're diabetic and trying to kind of balance out your glucose level. But what you need to know for this is there are some lipid breakdowns and protein breakdowns for cells, but again, the main source of energy for your body still would be carbohydrates. Because your brain doesn't store glucose as a main source of energy, and it requires the help of the body to receive that glucose, most of that glucose comes from protein through the process of gluconeogenesis. Second phase of starvation, your body taps into the fat as the main source of energy. That is where your body starts to convert the fatty acids into acetyl-CoAs, and again, ketone bodies. I did mention this on a previous lecture that ketone bodies are considered to be acidic, and therefore, as these ketone bodies are built inside your bloodstream, you will have a decline in your, or decrease in your blood pH, which makes your blood basically acidic. Ketone bodies also produce a sweet smelling breath, which is a really easy way of identifying if somebody is utilizing fat as a main source of energy, compared to a healthy individual, which will be tapping into glucose if they are eating normally and healthy. Your brain in the second phase of starvation can utilize ketone bodies and as well as some of the glucose to basically survive. The second phase of starvation can last for a few weeks. Again, during this phase, the most important thing you need to remember is that you're tapping into your lipids and what are the source of energy for your brain. Third phase of starvation is when you actually tap into your third reservoirs, which is basically protein. So you're relying on oxidative deamination of your amino acid. You will see again a lot of ketones because ketone body is also elevated. That's how you transport your acetyl-CoA. And during this phase, you have significant degradation of your skeletal muscles. Your protein resources for majority of the body is going to basically be tapped in as source of energy. So that can cause a lot of side effects. At this phase, this is really important. A lot of people basically will die in the third phase of starvation if they're not receive the nutrient they need, but they don't die. This is really important. They don't die as a result of low nutrition. They die because basically the immune system is shutting down, and as a result, they are very susceptible to secondary sources of infection as a result of that long-term starvation. So they don't die because they didn't eat food. They usually die because there is some sort of infection in their body. Last concept I want to talk to you guys about is associated with diabetes and patients who are diabetic. So they deal with a condition called diabetic ketoacidosis. So let me actually clarify a couple points for you guys first. When we're talking about a normal blood pH for an individual, if you are healthy and again it's maintained, we're looking at a number between 7.35 to 7.45, which is putting that on a slightly basic scale. If you're dealing with a ketoacidosis, your number is below 7.35, and if it's below 6.8, that will lead to death. So anything between 7.35 to about 6.8 is identified as acidosis. So what is exactly happening in a patient who hasn't been receiving its insulin? So if you look at this information, it says diabetic ketoacidosis occurs in people who are dependent on insulin. So they need to basically get daily injections of insulin. Now, why is that insulin important? We described this in our digestive system lecture. So insulin is basically allows the cells to uptake the glucose and use that glucose as a source of nutrients for your cells, right? Undergoing this whole process we described. So if you have no insulin in your bloodstream, just because either you didn't inject enough insulin or your body is not producing enough of it, so you are depending basically on the injection and you haven't received it, whatever the reason is, insulin is not present. So basically your cells, in another sense, cells don't have access to insulin. Don't have access to insulin, which means sorry, I can't spell apparently. Insulin. I'm going to rewrite this. They don't have access to insulin, which basically means their cell is low in glucose. Now, kind of imagine, what is your cell going to do when it doesn't have access to glucose? It's going to tap into your lipids. Now, lipids, when you process lipids, again, the most common one being triglyceride, you're going to produce your glycerol and fatty acids. They go through the process, they produce acetyl-CoA, and acetyl-CoA produces what? What we identify as ketones. What if when you have a lot of ketones in your blood? Ketones or ketone bodies are acidic. That causes a decrease in your blood pH. And again, that is what we identify as diabetic ketoacidosis. So why is that a concern? Why do I care if the blood pH is low? This is why it's important. And this is really important for you guys to understand as well, because these are essential for your exam. When you're looking at a blood pH of 7.35 to 7.45, which is your normal blood pH, that allows the oxygen to be effectively carried by your protein within your red blood cell called hemoglobin. Okay, so this hemoglobin carries the oxygen from your lungs into your tissues or into your cells. And when that when that oxygen gets into your tissue, hemoglobin releases that oxygen to your tissue, and your tissues are happy. So in your lungs, your hemoglobin is holding on to the oxygen because it wants to carry it. When it goes to your tissue, it's gonna drop off the oxygen and basically release it into your cells for utilization. When you drop your blood pH because you have a lot of ketones, your blood pH drops below 7.35. At that point, the affinity of hemoglobin for the oxygen drops throughout your body. This is really bad because it also drops in your lungs. So basically, instead of your hemoglobin holding onto the oxygen, is dropping off the oxygen. That causes shortage of oxygen inside your body. Your body doesn't have access to oxygen because hemoglobin is not carrying it very effectively anymore. Again, imagine no oxygen, your cells are going to be deprived of that ATP. No ATP, cells will eventually die. And this is where you're going to see organs being shut down because of lack of oxygen. So it's not the ketoacidosis that is causing the problem. It's the fact that these organs, other than the fact that, of course, that the blood pH is not optimal for many of the functions of the body, but it also affects the affinity of hemoglobin for oxygen carrying capacity, and therefore cells are going to be deprived of oxygen. So if you see a patient that is undergoing ketoacidosis, you see difference in terms of the pattern of breathing, because again, they are lacking that oxygen, so they're typically hyperventilating. You also kind of smell that sweet breath on them because of the high amount of ketones in their breath. If these patients are not treated, they will fall into a coma. And again, if you cannot manage their glucose or manage their ketone bodies and blood pH, it would lead to death for those individuals, unfortunately. So let's say this patient comes in, they are hyperventilating, you are smelling that sweet kind of a smelling breath on them, which is basically a sign of ketoacidosis. What is the treatment? One of the first things that you need to do is first increase their blood pH back to normal, which is best way to do that is using the buffer. We have talked about this again when we talked about acid and bases. That buffer is bicarbonate, which is written as HCO3 minus. And again, this is bicarbonate. So, but if I address the issue and add bicarbonate in terms of an IV to this individual, that's good. It may work for a little bit, but it's not going to address the underlying issue that caused this whole problem. So yes, giving the buffer and helping you maintain the blood pH is good, but it's not enough. So what is another thing you need to do? You need to give this patient insulin and glucose. Glucose is because basically you want to make sure that they stop producing ketone bodies, right? And insulin to make sure that they actually be able to bring that glucose into their cells and utilize it as a source of energy. So it's really important to make sure you combine glucose and insulin together, because if it's only one of them, it will not be able to be effective. So if you only give them insulin, they won't have access to glucose. And if you only give them glucose, they won't be able to bring them into their cells to utilize them as a source of energy. So if I ask you, what is the purpose of giving this patient glucose and insulin? Is for them to first take that glucose in and utilize glucose in terms of production of ATP, and therefore slow down the rate of ketone body formation, which helps you help bring your blood pH back to normal. That's about it. So that was the end of our lecture for metabolism and the end of the lecture for this semester. And again, make sure you guys are working on your homework, review the homework, and then let me know if you have any questions during our office hours. Thank you guys.